So are you noticing that you kind of are moving beyond Punnett squares? That you kind of don't need them with the stuff that we've been doing? Yeah. That's good, because that's where we're going. We are about to move beyond Mendel. What you've done so far is what is called Mendelian genetics. In other words, the stuff that Mendel was able to do with his limited knowledge, because we didn't know much at that point. We didn't know about DNA, chromosomes, any of that. So what Mendel knew, and it's pretty amazing he figured out what he did, is first of all, every organism gets a single copy of every gene from each parent. There's a gene for brown eyes versus not brown eyes. You got one of those, you got one version of that gene from your mom and one version of it from your dad. They might be different, they might be the same. That's what determines what you have on that gene. Yeah.
Does he make money from this research or something?
Well, he's dead. But when he was alive, I would say no, he did not make any money from this. He did most of this as a monk. And I'm guessing if any money came in from it at all, which I doubt, it would have gone to, the monastery.
What did he make on it?
Are these notes that we're taking today on the quiz tomorrow?
No. Good question. Yeah, this stuff starts our next unit. We're about to go into why does the stuff that Mendel figured out work the way it does. He figured out, okay, you get a single copy of every gene from each parent, so you've got two. And when your parents did that, when they gave you the single copy, they were able to do it because of the way they made their gametes. The way they made their sperm and eggs was a kind of weird cell division that splits the two alleles for every trait that they had and makes sure each new cell gets one, not both. So what this means is that if this is happening when gametes get formed, when sperm and eggs get formed, then sperm and eggs are weird. They must be like weird little half cells. Gametes are weird so that babies aren't. In other words, if you took two full cells with full sets of DNA and stuck them together, that's a funny looking kid. That's not going to work. They'd have double the DNA. We need to make little half cells so that when they come together, they make full cells.
Full DNAs connect.
In animals, it almost never happens. In plants, it does. When you see, and we'll talk about this, but when you see if you go to a county fair and you see the giant tomatoes or the fruits that are huge, that is actually a version called polyploidy of having a whole extra set of chromosomes. Yeah, animals don't survive it real well, but plants are a little more able to.
So if a person did that,
No, you just wouldn't see it because it wouldn't work because they wouldn't survive it. But yeah, theoretically, I guess. So moving beyond Mendel, we got to get to the early part of the 1900s, and this dude, his name's T.H. Morgan. And what Morgan did was start to connect genes that Mendel, theorized. Mendel said there must be genes. There must be these physical things that are making us this way. Morgan found them. He found where they were. And he did it because he found the perfect genetic test animal. He found the perfect guinea pig, which was not guinea pigs. For the perfect genetic test subject, what you need is the following things. You need something that doesn't live very long and has short generation time, so you can see lots of generations, within weeks or months rather than years. You need something that's real easy to control and contain. These animals you can keep in a jar, and they will live and eat and reproduce happily for you. And the final great thing about this organism turns out to be that if you take one and yank its head off, dangling from its head are these, I'll tell you what it is before you get weird, terrible nightmares. We're talking about fruit flies here, okay? Still gross, but yeah, I've done this. If you yank a fruit fly's head off, dangling from its head are two salivary glands. And I don't know how you figured this out, but if you separate the salivary glands, put them on a microscope slide and smush them with a cover slip, it'll bust the cells open. It'll bust the nuclei open. You got to heat them and stain them a little bit, but then what you see are the hugest chromosomes you've ever seen in your life. Fruit flies' salivary gland cells, for some reason, have gigantic chromosomes. And that's how Morgan was able to prove, basically, that, hey, genes are just places on chromosomes. Okay? So we think of him as the fruit fly guy, and he's the one that figures out, hey, when there's a band, when there's like a dark spot on this area of this chromosome, that fruit fly has red eyes. And if that band's not there, it has brown eyes. And he starts connecting properties of the chromosomes to actual physical traits of the fruit flies. Super cool. However, a lot of dead fruit flies. Like to think if there's a hell and T.H. Morgan went there, he's probably reincarnated as a fruit fly in his own lab. This always reminds me of my freshman in college lab partner, because we did have to do this. We had to take fruit flies and knock them out with ether, anesthetize them, lay them out on the table and track their traits. And then some of them we had to pull their heads off and actually look at their chromosomes. And my lab partner, who lasted about 2 quarters before he dropped out, was totally sniffing the ether. Don't do that.
You watched that movie, didn't you?
Yeah, that's what I think.
Yeah.
That movie wasn't out yet, but when I saw that movie, I absolutely thought of what's his name?
Yeah.
Really? Ryan. His name was Ryan. My lab partner.
And he just it's like it's not like
Buy fruit flies? Yeah. I mean, you order them. You order the eggs, I think, from like a biological supply company. Anyway, he figures out that it's chromosomes that are the places where genes live, where they are, and that organisms get 2 versions of every chromosome, not just every gene. You've got 2 versions of every gene because you have 2 copies of every chromosome. So while, yeah, you have 46 chromosomes, that's a little misleading because really what you have is 23 pairs. You have 23 chromosomes from your mom, and you have 23 from your dad. And any one of those that you pick out, say it's the mom version, you can find the dad version of that same chromosome, same genes, just maybe different versions, depending. So any chromosome you pick out in the nucleus, there's another one. And we use the word homologous. The chromosomes that are the same, but just have the versions from mom versus the versions from dad, those are your homologous chromosomes. And the process that Mendel figured out must exist, where those chromosomes get separated and put into sperm and egg cells, where you basically divide your chromosomes in half and make these weird little half cells. That process has a name, and that's the next place we're visiting. It's called meiosis. So next week, we'll get into what meiosis looks like, and the short answer is it looks a lot like mitosis, but with about double the number of phases. And it creates little half cells called sperm and eggs. Good. Be ready to do some genetics problems and answer some basic questions tomorrow for your quiz/test. Have a great day.